So, welcome guys. Starting with our crisp PYQ discussion of general pathology. So first question you can see Arthur's reaction. See, first trick I'll give you. So whatever may be the name of the reaction they are asking, whenever there's a naming reaction, mostly you can directly close your eyes and go with the idea that hack that it is going to be your type 3 hypersensitivity. So be it anything, okay, so be it anything, whenever there's a naming reaction, that should be your type 3 hypersensitivity. And you can see type 1 is IgE mediated, type 2 is your antibody mediated, type 3 is antigen antibody complexes. So like this only, one second. So like this only you should be thinking about it. Okay, so this is very important point, and most of your naming reaction comes under this thing only. And your type 4 is delayed type of hypersensitivity, that is T cell mediated. And your receptor against any receptor it is going, especially in Graves disease and Hashimoto thyroid disease, this and all, it comes under your type 5 hypersensitivity. So this is very important point. So Arthur's reaction is localized because we are having findings over our skin only. Okay, so next you can see this. So this is very important. So this disease is called as Bare lymphocytic disease. So what is this topic is about? So Bare lymphocytic disease is caused due to mutation in a transcription factors that is required for class 2. So for this thing you should be knowing two things, that is your class, what is class 1 MHC, class 2 MHC. Class 1 is your nucleated cell MHC, class 2 is your non-nucleated cells, or also is mostly your immune cells, your dendritic cells, antigen presenting cells, your macrophages, neutrophils. So these kind of cells will be having MHC2, and all your nucleated cells in the body will be having your MHC1. RBCs is non-nucleated, so that is going to not have your MHC1. So this is important. So in this thing, it's a case of complement deficiency, yes it can be, and CDT4, yes it can be, because immune mediated, right? MHC1, yes lymphocyte, yes nucleocytes, nucleated, so it is possible. But MHC2 on your lymphocytes, no, this is not. So MHC2, whenever that is going for problem, your complete immune system is going to shut off. So that is called as what? Combined immunodeficiency. Okay, so this is very important point. So next you are saying this HPV. So HPV E7. Okay, so this E7, it is going to inhibit your cyclin D, that is your P21 and P27. So your cyclin D1, that molecule is going to get inhibited. So whenever this is getting inhibited, your cyclin A and your cyclin C, so this is getting hyperactivated. So whenever this is why I am telling hyperactivated means that will result in uncontrolled proliferation of your cells, resulting in your carcinogenesis. So this is important. Here one inhibition is coming, here one activation is coming. This intermediate is your P21 and your P27. So this is important. So next coming to a toll-like receptor. So what are toll-like receptors? So just remember, toll-like receptor is a receptor, just a recognition receptors. It recognizes your specific protein sequences present on the surface of the antigens. Okay, so that will recognize it, and our body will elicit a specific type of immune response to it. So it is mediated by two things. One is your NFK beta pathway, and another one is what? Can you guess it? So this is nothing but your interferon mediated pathway. So one is your NFK beta pathway. This is asked in the PYQs, but your interferon mediated pathway is not asked in the PYQs. Okay, so NK cell marker. So NK cell marker, this is very important controversy. Whenever they are asking for a marker, it is your CD94. Whenever they are asking for surface receptor identification, go for 16 and your 56. Okay, its marker means 94, but surface receptor, it is your this thing. Okay, and which of the following is not seen in DIC? See, in DIC, everywhere the platelet is going to get activated, and all your coagulation clotting factors are going to get used up. So whenever that is getting used up, ultimately what will happen? Your prothrombin time will be increased. But here what is given? Decreased prothrombin time. So that is not going to happen. Okay, increased APTT, yes. Decreased fibrinogen, yes. Increased fibrin degradation products, yes. Obviously, that is the main sensitive marker, right? And also this is super important topic. Okay, this is very important topic. Why? So for this thing, you should be knowing only one hack. There are three categories of this proteins. Category 1 is our actin microfilaments, category 2 is our intermediate filaments, category 3 is our microtubules. Remember category 1 and 3. Under category 1 we are having G actin, F actin, category 1. Category 3 we are having microtubule, that is our tubulins, that's all. Vimentin, desmin, glial acidic protein, neurofilaments, everything, all the other names are going to come under your intermediate filaments. That's all you need to know. Okay, so next coming to your cell type and your major cytokine produced. So T cell 1, T helper cell 1. So T helper cell 1, gamma, mainly in your chronic inflammation, granuloma formation, your one cell is getting involved, gamma and interleukin 12. Okay, mostly for intracellular insult. That is why granuloma, you are having granuloma can be seen not extracellular, it will be intracellular. So macrophage activation and all will be seen IgG antibody because chronic inflammation, right? So this is how we should be linking it. And next coming to your Th2. So Th2, it is your 4-5-13. This is important. 4-5-13 aspect is important. And 4 for your parasites. All the parasitology completely dependent upon your Th2. Th17 will be your auxiliary. Auxiliary means it will be additional. So you should be remembering 17-22 for extracellular insults, mostly for your microbes or bacteria, neutrophils and monocyte responses mediated by this. So this is important. So the spotters. So why I have just kept this as spotters means, see, occupational lung diseases, everyone knows everything. But this histology aspect is very, very important in case of your INISET is concerned. So in this thing you can see anthracosis. So anthracosis is nothing but your pigment deposition. So this is a pigment deposition. You can see black pigment deposition. Here it is silicosis, means concretions. You can see the concretions here and there. Okay, wait, let me change the color. So concretions you can see here and there. Okay, so concretions you can see here and there. So this is important here. So next you can see this asbestos bodies also. You can see bodies, specific individual bodies can be seen here. Okay, so this is asbestos body, this is silicosis, and this is your asbestos anthracose. Okay, so this is important. And next you can see, wait, okay, so next you can see this next question. Yeah, so this all the immune deficiency disorders are important, but most important is this scenario, that is your rash plus platelet abnormality. So whenever this is going to happen, so whenever this is going to be there, you have to be VASP, that is your Viscott-Aldrich syndrome. Your VASP gene problem is going to be there here. Okay, that's all. Okay, so this is important. And next you can see this point. So that's what I was just trying to tell. And which are the following recognition and clearance? Yeah, this is very, very important aspect. So recognition is clearance is what they are asking. So in this thing, inflammasome, it for recognizing necrotic cell. Toll-like receptor, I already told you, it is recognizing molecules of pathogens. You shouldn't mess between these two. Okay. So next you can see this papain. This site is called as your papain site. The papain site, it is going to act and cut here. That results in your 2-Fab. This is a region of your antibody binding, and this is your FC region. Okay, so this is important. So that will result in your 2-Fab and your 1-FC papain binding. So next, alternative RNA splicing. So alternative RNA splicing is the most important thing, and it is a topic now. Why? Because there's all these similar molecules with the different fates, like your apolipoproteins, like your IgM, IgG, IgA, IgM, IgE antibody and all. So this kind of similar molecules with different subtypes. This main reason is because the main parent protein will be the same, the parent mRNA will be the same, but different RNA splicing, we are going to get different products. So this combination, B cell express different IgM and IgG body at the same time, is because of this alternative RNA splicing. The same mechanism is seen in apolipoproteins also. We have same apolipoprotein, same function, but different subtypes, different properties. Okay, so this spot is cell within a cell appearance. This is emperipolysis. So next, Castleman disease. Yeah, very, very important point. So whenever you are seeing histology with histopathology slide with multiple follicles, very, very hyperplasia, means that is your Castleman disease. Okay, so what is your Castleman's disease? First of all, 2 seconds. Okay, so Castleman disease is nothing but, it is justing but overgrowth of your own lymph nodes, nothing more than that. Okay, so overgrowth of your own lymph node, it can vary from, it's a rare disorder, it can vary from single lymph node involvement to multiple lymph node involvement, but here you can see single lymph node, every follicle is getting hyperplasia. So this is your Castleman disease. So next, coming to your mast cells. Mast cells, because of this granulation and histamine release, we are going to stain it with toluidine blue. Okay, and this is your Maltese cross, Fabry's disease. That's all. So these, you may wonder why will it be enough? You will definitely want to know, will it be enough for your general path? Okay, this is enough. Okay, this is enough, but not as a primary resource. Final revision material. That's all. Okay, good luck guys, with love. Thank you.